Welcome to your personal deep dive. Today we're going on a quest, kinda, to understand the language of life itself. DNA. You got it. We'll be exploring how those instructions, all those A's, T's, C's, and G's get turned into you, me, everybody. You provided some awesome research on the genetic code and transcription, including a textbook chapter summary. So get ready. We're about to unlock some serious secrets of your own biology. How about that?
It's truly amazing. It really is mind-boggling how this tiny microscopic code orchestrates everything. I mean, the whole symphony of life.
Right.
We'll be deciphering this language, and we'll be exploring how DNA's message gets transcribed into RNA and then ultimately translated into the proteins that build and run your body.
Yeah, so it's like, DNA holds the blueprints to you. But how does it actually, guide the building process? It's not like there are, tiny construction workers in our cells running around with, little hammers and nails.
You're right. There are no tiny hammers and nails.
Okay, good.
Instead, think of DNA as a master blueprint, okay? But it's stored securely in a vault. We don't want to mess with that original, right?
Right.
So to actually build something, you need a working copy.
Okay.
And that's where RNA comes in. RNA is like the messenger. It carries those instructions out to the construction site, and that construction site is the ribosomes. And that's where proteins, the real builders, they get to work.
Okay.
This whole process, DNA to RNA to protein, it's called the central dogma of molecular genetics. It's a big deal.
Central dogma. So DNA is the architect, RNA is the contractor, and proteins are the construction crew.
I like that.
I like that analogy. And what's really fascinating is that this system, this whole DNA to RNA to protein, it's practically universal, right?
Yes, it really is. From the tiniest bacteria to us, to humans,
Yeah.
almost all living organisms use this same fundamental code.
Wow.
There are a few intriguing exceptions, but for the most part, everybody's doing it the same way.
That's amazing.
Yeah, and it highlights how interconnected life is at the molecular level. Like, we're all in this together.
We are all in this together. So let's dig in. Let's delve into the code itself, the DNA code.
Okay.
How does it actually work?
Well, DNA's alphabet, it only has 4 letters, right?
Right.
A, T, C, and G. And those represent the chemical building blocks of DNA. But these letters, they combine to form 3-letter words, and we call those codons.
Okay. So we got a language with 3-letter words. That's pretty efficient, right?
Very efficient.
But how does that build something as complex as, well, us?
Yeah.
We're pretty complex organisms.
Well, each of those 3-letter codons, it corresponds to a specific amino acid.
Okay.
Amino acids are the building blocks of proteins, and there are 20 different kinds. So these amino acids can be combined in countless ways to create all the different proteins in your body, and each one has its own unique function.
So it's like each codon is a recipe instruction for adding a specific ingredient to our protein soup.
I like it.
And the possibilities are endless.
Endless, that's right. But here's where it gets even more interesting. Okay, so there are 64 possible codons, right? Because you have 4 letters and you're making 3-letter words. There's a lot of different combinations.
Right.
But there are only 20 amino acids. So this means that some amino acids are specified by multiple codons.
Oh, okay.
Yeah.
So there's built-in redundancy. Like, is there a reason for that? It's like, why have multiple codons for the same amino acid?
Well, this redundancy, it's not just a fluke. It's like a brilliant safeguard.
Okay.
Think about it. If there's a typo in the DNA blueprint, and, let's face it, mistakes happen.
They do, yeah.
This redundancy can actually prevent a catastrophic error in the final protein.
Okay.
It's like having a built-in spell checker.
Oh, nice.
For our genetic code. So even if there's a little mistake, a lot of times it's not a problem.
That's incredible. A safety net built right into the system.
It's pretty cool.
Yeah. So, and just like any good language, you got to have rules about where sentences begin and end, right?
Absolutely.
Like punctuation.
Yeah. Just like punctuation marks in a sentence, we have specific start codons and stop codons.
Okay.
These signals tell the cell where to begin and end protein synthesis.
Okay.
And it ensures that the proteins are made with the correct sequence of amino acids, which is essential for their function.
Right. So we have our blueprint, and we're starting to understand the language.
Right.
But how do we actually get from DNA to RNA? That's where transcription comes in, right?
Precisely. Transcription is the process of creating that working copy of the DNA instructions. That's the messenger RNA, or we call it mRNA for short.
mRNA, okay.
And it's kind of like, it's like carefully copying a recipe from your favorite cookbook onto an index card so you can take that into the kitchen without risking damage to the original. You don't want to spill anything on that cookbook.
No, keep the cookbook nice and clean.
Exactly.
So the chef in this scenario would be?
The chef is an enzyme called RNA polymerase.
Okay.
And it binds to the DNA. It unwinds the double helix. Remember, DNA is a double helix. And then it meticulously reads the DNA sequence, and it assembles a complementary RNA strand as it goes along.
Okay, so RNA polymerase is like a molecular scribe.
I like it.
Transcribing the genetic code from DNA to RNA. But how does it know where to start and stop copying? You know, like, are there signals?
That's a great question. That's where promoters come into play.
Promoters?
Yeah. These are specific DNA sequences,
okay.
and they act like start here signs for RNA polymerase.
Okay, so they're like road signs.
Exactly. And they tell it where to begin transcribing. And they can also control how often a gene should be transcribed.
Oh, okay.
So, like, some genes need to be transcribed a lot, others maybe not so much.
Yeah.
Promoters help regulate that.
So promoters are like recipe titles.
Okay.
That tell the chef which dish to prepare.
I like it.
But what about stopping?
Right.
We wouldn't want a giant blob of RNA, would we? Like, where does it stop?
No, we don't want a giant blob of anything.
Right.
The transcription process, it's divided into 3 stages. You have initiation, elongation, and termination.
Okay.
It's a bit like a well-choreographed dance.
Okay.
So first you have initiation, RNA polymerase binds to the promoter, and that starts the whole process of transcription. Then you have elongation, RNA polymerase moves along the DNA, creating the mRNA copy.
Okay.
And then finally, termination, when it reaches a specific signal in the DNA sequence, it detaches, it lets go, and that releases the completed mRNA molecule.
Okay.
3 steps.
Nice. A molecular ballet.
Right.
That's cool. But we've been talking about bacteria.
Right.
Isn't transcription more complex in organisms like us? Like eukaryotes?
You are absolutely right. Eukaryotic cells, like the ones that make up you and me, they have a more elaborate process.
Okay.
For one thing, we don't just have one RNA polymerase, we have 3.
Oh, wow. Okay.
3 different ones, each specializing in transcribing different types of RNA.
So we have specialists.
We have specialists, yeah.
For different tasks.
Yeah.
Okay.
It's a division of labor.
Right, but hold on. Isn't our DNA packaged differently too? Like, it's not just floating around in the cell willy-nilly.
You're thinking about chromatin.
Chromatin.
Yeah. So our DNA, it's tightly coiled around these proteins, and that forms this structure called chromatin.
Okay.
And before transcription can even begin, this chromatin needs to be unwound. Because it's so tightly packed in there, it has to be made accessible to the RNA polymerase.
So imagine trying to read a recipe that's rolled up really tightly.
Exactly.
You need to unroll it first.
You unroll it, yeah.
You can't just go in there and start reading.
No.
So first we unpack the DNA, then the right RNA polymerase needs to find its promoter, its little road sign, and then it can start transcribing. Okay, anything else that makes eukaryotic transcription unique? Anything else special?
Absolutely. So eukaryotic mRNAs, before they can be used to actually make proteins, they go through this makeover.
Oh, a makeover. Tell me more.
Yeah. They receive a special cap at the beginning.
A cap.
And a tail at the end. And both of these, both the cap and the tail, have very important jobs.
A cap and a tail. What's the purpose of those? What do they do?
So the cap, it's kind of like a protective helmet for the mRNA.
Okay.
It helps prevent it from being degraded too quickly.
Right. Because
the cell is a very busy place.
Yeah, things are getting broken down and rebuilt constantly.
Exactly. And you don't want your important message, your mRNA, to be destroyed before it can do its job.
Right. So the cap protects it.
The cap protects it.
Okay. What about the tail? What does the tail do?
The tail, it acts like a signal flare.
Okay.
It helps direct the mRNA to the ribosomes.
Okay. So it's like saying, hey, over here, come translate me.
Come translate me, yeah.
Okay, so we got the cap, we got a tail. What else is happening in this mRNA makeover?
Well, remember how we talked about introns?
Introns, yeah, the non-coding regions.
The non-coding regions.
It's like the bloopers in the film.
Exactly.
Right.
So eukaryotic mRNAs, They have to go through this process called RNA splicing.
RNA splicing, and that's where those introns get cut out, right?
That's where the introns get cut out, yep.
Okay.
And the remaining pieces, those are the exons, remember?
Yeah, exons.
The coding sequences, they get stitched back together.
So it's like we're literally editing.
We are literally editing.
The RNA molecule.
Yes.
Yeah.
It's a very precise process.
So how does the cell know which parts to cut out and which parts to keep?
That's a great question. There are specific sequences at the boundaries between the introns and the exons that act like signals for the splicing machinery.
And the splicing machinery is?
The splicing machinery is a complex called the spliciosome.
The spliciosome? It sounds like something out of a sci-fi movie.
It does, doesn't it?
Yeah.
It's a large and complex molecular machine made up of RNA and protein components, and it recognizes those specific sequences at the intron-exon boundaries, brings them close together, and then it catalyzes the cutting and pasting reactions that remove the intron and join the exons together.
It's mind-blowing.
It is.
How these tiny molecular machines can perform such complicated tasks with incredible precision.
Right.
It's amazing.
It really speaks to the efficiency and accuracy of the cellular world. It's a testament to how incredible it all is.
Yeah, and we talked about alternative splicing too.
Yes.
Where different proteins can be made from the same gene. Like, how does that work? Does the spliciosome just decide to like skip over certain exons sometimes? Like, is it random?
Well, it's not totally random.
No way.
The spliciosome itself doesn't like decide, but it can be influenced by these regulatory proteins that bind to the RNA molecule.
Okay.
And these proteins can either promote or inhibit the splicing of particular introns.
Oh.
So that leads to different combinations of exons in the final mRNA.
So it's like having a director giving the film editor instructions on which scenes to keep or cut to create different versions.
Exactly. Different versions of the movie. That's a great analogy.
Of the movie, yeah.
Yeah.
Okay, and that's important because it increases the diversity.
Yeah, it dramatically increases the diversity.
Proteins our body can make. Okay, so it's like we have a basic recipe that we can tweak and modify to create like countless variations.
That's right.
I'm starting to understand why our bodies are so complex.
It is, yeah.
It's incredible.
It's all thanks to that flexibility of our genetic code.
It's amazing. So we've got DNA, we've got transcription, we've got RNA splicing, we've got alternative splicing.
Right.
Is there anything else we need to know about this incredible journey?
Well, there's one more fascinating aspect that I think we should discuss, and that's RNA editing.
RNA editing. So it's not just cutting and pasting. Like, we can actually like rewrite parts of the script.
Precisely. So RNA editing it's another way to change the genetic information that's encoded in the DNA.
Okay.
But instead of happening at the DNA level, it happens after the RNA molecule has been transcribed.
Oh.
So it's like making edits to the movie script even after the filming's been done.
Okay.
Yeah. And there are a couple of different ways that RNA editing can occur. Sometimes individual bases in the RNA sequence, they're chemically modified. And these seem like small alterations, but they can actually change which amino acid gets added to the protein.
Oh, wow.
Potentially altering its function.
So it's like changing a single letter in a word and creating a whole new word with a different meaning.
Exactly. It can have a big impact. It can have a big impact, yeah.
So, for example, think about the editing of apolipoprotein B mRNA in your intestinal cells. A single change from a C to a U creates a stop codon. And that results in a shorter version of the protein. So it just shows how even a tiny edit can make a big difference.
Big difference.
Yeah.
Wow, it really is about the detail.
It is. It is.
But you said there were other types of RNA editing too?
I did. Sometimes nucleotides, those are the building blocks of RNA. They're actually inserted or deleted from the RNA sequence. And this process can be quite extensive in some organisms. For example, trypanosomes. Yeah, these are parasites that cause sleeping sickness. They use RNA editing to dramatically diversify their proteins.
So it's like adding or deleting whole words or even sentences.
Script. From the script, right?
Wow.
And this process, it's not random either.
It's guided by these small RNA molecules called guide RNAs or gRNAs.
gRNAs.
Yeah. And these molecules act like templates. Okay, so they direct the editing machinery to make specific changes.
So it's like having a team of expert editors and proofreaders
Right.
meticulously refining
the script.
the script to perfection.
Yeah. And this RNA editing process, it adds yet another layer of flexibility to the genetic code. So it allows for even greater diversity and adaptability in living organisms.
Yeah. This is incredible.
Yeah.
We've come a long way from just thinking about DNA
Yeah.
as, the blueprint of life.
Yeah. It's so much more than that.
Yeah, it's a whole process, a whole system.
Absolutely.
But wait a minute.
Mm-hmm.
Back in the beginning, you mentioned there were some exceptions to this universal genetic code.
You're right.
Like, what are those? Do some organisms not follow these rules?
So while the genetic code is remarkably consistent across most organisms,
Yeah.
there are a few outliers. These exceptions, they're particularly prevalent in mitochondria.
Mitochondria, the powerhouses of the cell.
The powerhouses, that's right.
What makes them so special?
Well, for one thing, they have their own DNA,
Oh, okay.
separate from the DNA that's in the nucleus.
Okay.
And their genetic code, it's evolved a bit differently.
So it's like they're speaking a slightly different dialect.
Yeah, different dialect.
Of the genetic language.
A great way to put it.
Yeah. Okay, give me an example.
Okay, so for example, in human mitochondria,
Mm-hmm.
the codon UGA,
Okay.
which is usually a stop codon in the standard genetic code,
Right.
it actually codes for the amino acid tryptophan.
What?
Yeah, so what's normally a period in the genetic sentence
A full stop.
is actually a word in mitochondrial language.
That's fascinating. Why would that be? Why would it be different?
Well, it's thought to be a result of their unique evolutionary history.
Okay.
Remember, mitochondria, they were once free-living bacteria
Oh, right.
that were engulfed by early eukaryotic cells.
Billions of years ago.
Billions of years ago.
Yeah.
And over time, their genetic code, it's retained some of its kind of ancestral features.
So it's like finding these ancient inscriptions that give us clues
Yeah.
about the history of language.
I like that. A wonderful analogy.
Yeah.
Yeah, and these exceptions to the genetic code, they remind us that evolution is a continuous process. It's constantly exploring new possibilities.
That's amazing.
Yeah.
So this deep dive has truly opened my eyes
Good.
to the incredible complexity and flexibility of the genetic code. It's like peeling back the layers of a fascinating mystery, like a detective novel.
It is. It's a good mystery.
Yeah.
And it's a testament to the power of evolution.
Yeah.
To create such a robust and elegant system for storing and expressing genetic information.
I have to admit, after all this talk about transcription and RNA splicing and editing, I'm feeling a little overwhelmed.
It's a lot, I know.
Yeah, it's like trying to keep track of all the characters and plot twists in a complex movie.
It is a lot to take in.
Yeah.
But remember, the key takeaway here is that all of this intricate system, it serves one purpose, and that is to translate the information that's encoded in your DNA into the proteins that carry out all the essential functions of life.
Right, that helps to put it all into perspective.
Yeah.
Okay.
It's all about making proteins.
Making proteins.
That's what it's all about.
Okay, that makes sense. So where do we go from here? What happens next in this whole process?
Well, I think now we need to move on to the next stage,
Yeah.
which is translation.
Translation.
Yeah. So we've talked about how the information gets encoded in DNA,
Right.
and then how it gets transcribed into RNA. But now we need to talk about how that RNA actually gets used to build proteins.
Okay, sounds like a plan. Let's jump into translation.
Let's do it. Welcome back to our dip dive. We're still uncovering all the mysteries of the genetic code.
Yeah, we just finished discussing how eukaryotic cells give their mRNA like a makeover, right?
Yeah, they get a cap and a tail.
Yeah, the cap and the tail.
Right.
But before those mRNAs can be used to actually build proteins, they go through another crucial process. It's called RNA splicing. And this is where those mysterious introns that we talked about earlier come into play. The introns.
The introns, yeah.
So we have this, this long RNA molecule with all these non-coding introns
Right.
scattered throughout, and the cell has to cut them out and then perfectly stitch the remaining pieces, the exons, back together.
Yeah.
It seems like, I don't know, a really risky surgery for a tiny cell to be doing.
It does. It's like a film editor.
Okay.
Meticulously removing all the unnecessary scenes, all the bloopers, and then seamlessly joining the important footage together.
Right?
To create a coherent and meaningful story.
But how does the cell know which parts are the bloopers and which parts are, like, crucial for the story? It all seems incredibly complicated.
Yeah, so there are specific sequences
Okay.
at the boundaries between introns and exons. And these sequences, they act like signals for the splicing machinery. And these sequences, they're highly conserved, meaning they're very similar across different genes and organisms. So it's like a universal language that tells the cell, cut here.
Cut here. Okay, so it's like having these specific markers in the film that tell the editor exactly where to make the cuts.
Exactly. And the machinery responsible for carrying out the splicing, it's called the spliceosome.
Spliceosome. Again, sounds like something out of a sci-fi movie.
It does. It does. It's a large
Yeah.
and complex
Yeah.
molecular machine
Okay.
made up of RNA and protein components. And this remarkable machine, it recognizes those specific sequences at the intron-exon boundaries, brings them close together, and then catalyzes the cutting and pasting reactions that remove the intron and join the exons.
It's mind-blowing how this tiny molecular machine can perform such a complicated task.
I know.
With such incredible precision.
It is amazing.
But earlier we talked about alternative splicing,
Yes.
where different proteins can be made from the same gene.
Mm-hmm.
Like, how does that work? Does the spliceosome just, I don't know, decide to skip over certain exons sometimes?
Well, the spliceosome itself doesn't decide.
Okay.
But it can be influenced by these regulatory proteins that bind to the RNA molecule.
Okay.
And these proteins can either promote or inhibit the splicing of particular introns,
Okay.
leading to different combinations of exons in the final mRNA.
So it's like, having a director
Uh-huh.
giving the film editor instructions on which scenes to keep or cut
Okay.
to create, like, different
Versions of the movie, yeah.
the movie.
Yeah.
Exactly.
Okay.
And this alternative splicing it's incredibly important because it dramatically increases the diversity of proteins that your cells can produce from a limited number of genes. So it's like having like a basic recipe, like a cake recipe,
Right.
but you can tweak it
Mm-hmm.
to create like countless variations, like
Okay.
chocolate cake, vanilla cake, strawberry cake,
That's right. All from the same basic recipe.
Yeah.
You just tweak it a little bit.
I'm starting to understand why our bodies are so complex.
Yeah.
Like, there's so much going on.
There is.
It's incredible.
It's all thanks to that flexibility of
Yeah.
the genetic code.
That's amazing.
Yeah.
Okay.
Right.
So we've got DNA,
Mm-hmm.
we've got transcription,
Yeah.
we've got RNA splicing,
Uh-huh.
we've got alternative splicing.
Right.
Is there anything else we need to know about this incredible journey from genes to proteins?
Well, there's one more fascinating aspect that I think we should discuss, and that's RNA editing.
RNA editing.
Editing.
So it's not just cutting and pasting, but like we can actually rewrite parts of the script.
Precisely. So RNA editing offers another way to change the genetic information that's encoded in DNA. But instead of happening at the DNA level, it occurs after the RNA molecule has been transcribed.
So it's like making edits to the movie script even after filming is wrapped up.
Exactly. And there are a couple of different ways that RNA editing can occur. Sometimes individual bases in the RNA sequence are chemically modified, and these seem like small alterations, but they can actually change which amino acid is added to the protein, potentially altering its function.
It's like changing a single letter in a word and creating a whole new word with a different meaning.
Exactly. It can have a big impact.
Yeah.
So, for example, think about the editing of apolipoprotein, B mRNA, in your intestinal cells. A single change from a C to a U creates a stop codon.
Okay.
And this results in a shorter version of the protein. So it just highlights how even a tiny edit can make a big difference.
Wow. It really is about the details.
It is. It is.
But you said there were other types of RNA editing too.
I did. Sometimes nucleotides are actually inserted or deleted from the RNA sequence. And this process, it can be quite extensive in some organisms.
Okay.
For example, trypanosomes.
Trypanosomes, those are parasites.
Yeah, parasites that cause sleeping sickness.
Yeah.
They use RNA editing to dramatically diversify their proteins.
So it's like adding or deleting entire words or even sentences from the script.
Or even sentences, yeah.
Wow.
And this process, it's not random either. It's guided by these small RNA molecules called guide RNAs or gRNAs.
gRNA.
And these molecules act like templates, okay? So they direct the editing machinery to make very specific changes.
So it's like having a team of like expert editors and proofreaders, meticulously refining the script.
Refining the script, exactly.
To perfection.
Yeah.
Okay.
And this RNA editing process, it adds yet another layer of flexibility to the genetic code. So it allows for even greater diversity and adaptability in living organisms.
This is incredible. We've come a long way from simply thinking about DNA as the blueprint of life.
It's so much more than that.
Yeah, it's a whole process, a whole system.
The whole system, yeah.
With all these amazing checks and balances.
Checks and balances, that's right.
And backups and just incredible complexity.
Portable complexity, yeah.
It's mind-boggling.
It really is.
Okay, so we've talked about DNA, we've talked about transcription, we've talked about RNA splicing, alternative splicing, RNA editing. Is there anything else we need to cover before we move on to the final step, translation?
I think we've covered the major points. We've really delved into the complexities of how genetic information is manipulated and regulated from DNA to RNA. And now I think we're ready to explore the final stage where that messenger RNA is actually used to build the proteins that carry out all the essential functions of life.
Sounds good.
Yeah.
Let's move on to translation. Welcome back to the final part of our deep dive into the genetic code.
We've journeyed from the depths of DNA structure all the way to the complexities of transcription, RNA splicing, and even RNA editing.
Yeah, it's been quite a ride through the molecular landscape of our cells.
It has. And now we finally arrive at the culmination of this whole intricate process, translation.
Okay, so this is where the blueprint finally comes to life.
Precisely. If DNA is the architect's grand design and mRNA is that carefully crafted blueprint, then translation is the actual construction phase where the skilled workers, they bring that design into reality.
And who are these skilled workers in our cellular construction site?
Well, the key players here are ribosomes.
Ribosomes?
Yeah. These remarkable molecular machines, they act as protein synthesis factories, churning out all the proteins your body needs.
So ribosomes, we've heard that name before. They're made of RNA too, right?
Yes. Ribosomes are composed of ribosomal RNA, or rRNA.
RNA.
Along with various proteins.
Okay.
And they're found in the cytoplasm of all living cells. And their job is to read that mRNA sequence and then assemble the corresponding amino acids into a polypeptide chain. And eventually that folds into a functional protein.
So if mRNA is the instruction manual, ribosomes are the skilled laborers reading those instructions and assembling all the building blocks.
A perfect analogy. Now let's break down this process a bit further. The mRNA molecule carries a series of codons, those three-letter words we talked about earlier,
right,
that specify which amino acid should be added to the growing protein chain.
Okay, so we learned about codons in part 1.
We did.
But how does the ribosome actually know which amino acid goes with which codon?
That's where another type of RNA comes in, transfer RNA, or tRNA.
tRNA.
And each tRNA molecule, it has two crucial parts. It has an anticodon, which is at the 3-base sequence that's complementary to a specific mRNA codon.
Okay.
And then it has an attachment site for the corresponding amino acid.
So it's like each tRNA is a specialized delivery truck carrying a specific building material and a label that matches the instructions on the blueprint.
Precisely. And the ribosome acts as this platform where the mRNA and the tRNAs, they all come together.
Okay.
The ribosome reads the mRNA codons one by one. And for each codon, the matching tRNA carrying its specific amino acid binds to the ribosome.
So it's like a perfectly choreographed assembly line with each worker adding their piece at just the right moment.
And as each tRNA delivers its amino acid, the ribosome actually catalyzes the formation of a peptide bond between the newly arrived amino acid and the growing polypeptide chain.
So the ribosome isn't just a platform, it's like the foreman, overseeing the construction and making sure all the pieces are joined together correctly.
Exactly. And this process continues codon by codon, amino acid by amino acid, until the ribosome hits a stop codon on the mRNA.
The stop codon, the signal to end construction.
Yes. When the ribosome encounters a stop codon, there's no matching tRNA.
Okay.
Instead, a release factor, it binds to the ribosome.
Yeah.
And this triggers the release of that completed polypeptide chain.
So the construction crew packs up, and we have our finished protein ready to carry out its function in the cell.
Exactly.
It's amazing how this all happens, like flawlessly, you know?
It really is. It's a remarkable process, and it happens countless times every second in every cell of your body.
That is incredible.
Yeah.
So we've gone from DNA to RNA to protein. Is that the end of the story?
Well, not quite. You see, newly synthesized proteins, they're often not functional right away.
Right.
They need to fold into these specific three-dimensional shapes
Okay.
to be able to do their jobs.
So it's like after the building is constructed,
Yeah.
it still needs to be furnished and decorated to become a functional home or office or whatever it's going to be.
A great analogy.
Yeah.
And just like an interior designer carefully arranges furniture and decorations, we have these specialized proteins called chaperones
Chaperone.
that help newly synthesized proteins fold into their correct shapes.
So chaperones are like the finishing crew, making sure everything's in its right place.
Right.
And the final product is functional.
Exactly. And sometimes proteins also undergo further modifications, like the addition of sugar molecules or phosphate groups.
Yeah.
These modifications can, fine-tune their activity and even determine their location within the cell.
So it's like adding those final touches,
Yeah.
like a fresh coat of paint or some landscaping to make the house truly complete.
I like it. And with that, we've reached the end of our journey from DNA to protein.
This deep dive has been incredible. Really, like an incredible exploration of this amazing molecular machinery that drives life.
We've seen how genetic information is faithfully transcribed from DNA to RNA, processed, refined through splicing, editing, and ultimately translated into the proteins that carry out all the essential functions of our cells, your cells, everybody's cells.
It's just amazing. It's a testament to the power of evolution to create such an elegant, robust system for building and maintaining life.
And it also highlights the interconnectedness of all living things, from the simplest bacteria to us complex multicellular organisms. We all share this fundamental process of genetic expression.
Yeah, we do.
It's pretty remarkable.
I'm left with a profound sense of awe and wonder at the complexity and the beauty of the molecular world.
As am I. Hopefully you've gained a deeper appreciation for that intricate dance of molecules that makes life possible.
I have. This has been an amazing deep dive. We've uncovered so many secrets of DNA's language. We've explored the intricate processes that transform this code into the building blocks of life.
It's quite a journey.
It is. But we've also learned that this journey doesn't stop here.
It doesn't.
There's always more to explore.
Always.
Absolutely. And as we discovered, the flow of information isn't always a one-way street.
Right.
Changes in the environment or even our lifestyle choices can sometimes influence our genes, which is incredible. Definitely a topic for another deep dive. But until then, thanks for joining us on this incredible journey of discovery.
It's been my pleasure.